And in my work, I've mostly focused on dopamine-related processes, and you might know that dopamine is involved in motivation and cognitive control, and we're now starting to understand that it's also really involved in the crossover between motivation and cognition. So is something worth the effort for the reward that you get out of it? And interestingly, aging is accompanied by a decline in dopamine signaling and also by impairments in reward anticipation, cognitive control, executive functioning like working memory, and also by a reduced willingness to actually expend effort for cognitive effort for a reward. So what we've done in the past is give a dopamine precursor, which is in tyrosine, which is an amino acid, to 29 older adults. And we found that giving this dopamine precursor versus placebo enhanced the willingness to invest cognitive effort for reward. We only found this in low impulsive subjects, which is due to the task. So why this clearly shows the role of dopamine in this type of cognition, but why is this actually interesting is that now, recently we've also done in healthy young adults an acute inflammatory challenge. LPS was also mentioned in yesterday's interesting session about inflammation. So this was, yesterday was mentioned in rodents. We can also give this to humans. These are students who are in a medium care facility. They get LPS intravenously and they'll show sickness behavior, which was also mentioned yesterday. So they feel fatigued and they have altered motivational priorities. So interestingly, there's a lot of studies being done on physical effort, and we have now recently shown that LPS, or an acute inflammatory challenge, also reduces the willingness to invest cognitive effort without really affecting reward per se. And in fact, brain dopamine might be particularly suited to signal those metabolic demands from the body and to really drive these to shape this effort allocation or these motivational priorities. And this is, for instance, because of it signals like the hunger and satiety, peptide hormones signal to the dopamine system whether to go for a food reward or not. And also it's well known that inflammation affects the brain dopamine system to whether to relax and not be too busy and active when you're ill. And of course, if we look at inflammation, we also think about the gut microbiome. And I won't go into this whole schematic of Michael Treadway and colleagues, but they've shown that even things associated with low-grade systemic inflammation through various pathways might reach the brain and then affect dopamine signaling by, for instance, reducing dopamine production. As we all know, inflammation and gut microbiome are also affected in aging, so it would be interesting perhaps to look at those mechanisms in aging. Well, I first would like to show you some work that we've done in young adults, but I'll also show you some plans for in cognitive aging. So when we talk about inflammation, I'm also very interested in the gut microbiome brain axis, which is of course influenced by diet, and which can have its effect through the immune system, as mentioned, but also through producing all kinds of metabolites, which are also neurotransmitters and its precursors, which can actually cross the blood-brain barrier and perhaps affect neurotransmitter production in the brain, like these precursors of dopamine. So our question is, can we use functional neuroimaging to show the relation between the gut microbiome and the brain? And here's a schematic overview with the reasoning. So we reasoned that the gut microbiome can synthesize these precursors of dopamine, like phenylalanine and tyrosine, and which might cross the blood-brain barrier and affect dopamine-related processes. So do differences in microbial dopamine synthesis, precursor synthesis, relate to differences in brain responses for reward anticipation, which is a typical dopaminergic function which you can readily measure through with functional neuroimaging in a very objective way. So we had participants going into the scanner doing a reward anticipation task, so they made effort to gain a monetary reward. And we also had them, we collected stool samples from them. And we used 16S rRNA sequencing to predict their dopamine precursor of the complete gut microbiome, the dopamine precursor synthesis. So this is really, I should say, this can be better done with shotgun metagenomics, which is more expensive. So this is, it's important to note that this is just a prediction based on a database PyCrust. And these are the collaborators who know a lot more about the gut microbiome. And well, first, this task clearly activates the ventral striatum as we expect. So this is for high versus low reward cues. And now, based on a certain pre-selection, which I won't go into detail here, but we found one bacterial enzyme related to phenylalanine synthesis, CDT. And this one was indeed related to ventral striatal activity for reward. So a microbial enzyme involved in the production of a dopamine precursor was related to brain responses during reward motivation. I must say it was a small study, it was a correlational study, it was based on a prediction, but this was really for us to show that we can look at these functional gut microbiome measures and relate them to brain function. So in the next step, we wanted to see a bit more of a causal relationship using the intervention, and we wanted to look a bit more at cognitive control. So this is with a multi-species probiotics intervention, and this was a probiotic designed to protect the epithelial barrier. And our main primary question was whether we could see effects in brain and cognition, and a secondary objective was to see whether these effects could perhaps depend on stress, since stress is known to negatively affect the epithelial barrier. And this was in healthy young female students. And this work was done by Silvia Papalini. And we compared an 8-week multi-species probiotics with a group who got a placebo. And the participants, before and after, went into the scanner, did a cognitive control task. And they also did a behavioral working memory task before and after a acute stress challenge, which was a socially evaluated cold pressure test. Well, first, for the cognitive control task, as expected, we nicely see frontal striatal regions in this task, and this was all well, but we didn't see any effect of the probiotics in this healthy young group. So we were a bit surprised. This was our main objective. Looking at the literature is perhaps not so surprising because these were very healthy students and they had really nothing to improve. We also looked at mood measures, some emotional scanner tasks, which have been used a lot in probiotics research. But our group was really healthy, didn't have any depressive complaints, so we couldn't see anything here. Then we looked at the stress task, so working memory before and after stress. And here on the y-axis is the difference. If the bar goes down, there's a detrimental effect of stress, and indeed in the probiotics group, before taking the probiotics, there was this detrimental effect of stress on working memory as expected. And after taking the probiotics, this effect disappeared. What you might already notice that in the placebo group, we could not see this detrimental effect of stress so clearly, although this effect was only trend significant, this baseline difference, this should still be interpreted with caution. But it does seem to indicate that there is perhaps a role for probiotics in a buffer to stress-induced disruptions in cognitive control. But given these trend baseline differences, we were also interested in individual differences. So we were looking at how those same participants that got a benefit in this stress-induced working memory by the probiotics, how did their brain change during the cognitive control task that they did before this stress task. And here you see that clearly in the only in the probiotics group, the participants who had a bigger benefit of the probiotics on the stress-induced working memory also were more efficiently recruiting their prefrontal cortex. And after correcting for the number of regions of interest, we also see that this was significantly different from the effect in the placebo group, where we didn't find such a relation whatsoever. So we did see a relation between probiotics-induced probiotics-induced benefit in stress-related working memory and the prefrontal cortex recruitment. Now, in a later stage, we got some more money to actually look at the stool samples of those participants, and this was done by Mirjam Bloemendaal. And here we could see that after another pre-selection method, we found, after correcting for multiple comparisons, that there was one genus, an unclassified genus in the Ruminococcaceae family, that was related to this beneficial effect in stress-related working memory. So the participants who benefited the most were also the ones who had the most probiotics-induced related increase in this genus in the Ruminococcaceae family. But of course, it would have been really interesting to see what the function was instead of focusing on this one genus, but we didn't have the means to look at that in this study. So in this healthy young sample, we only found beneficial effects of the probiotics on cognitive control following acute stress, and this effect related to changes in the PFC and in a Ruminococcaceae genus. And now the interesting thing would be to look in an aging population, because they already have alterations in the gut microbiome, so they might benefit from certain nutritional or lifestyle interventions via the gut microbiome or via inflammatory processes. And I won't have to explain this to you, that perhaps a multi-domain intervention might have the biggest effect on these processes like Neuroimaging. So I'm happy to present to you the MOCIA project, which is a public-private consortium from the Netherlands with 8 fantastic Dutch universities and 8 fantastic public-private partners. Some of them are here, and I'm very happy to lead this consortium. I will focus mostly on the mechanistic neuroimaging work package, but I'll take you a bit through the other work package as well. So the first one, you might have already heard about it, is the finger-NL work package, where we do a finger-type multi-domain lifestyle intervention in more than 1,200 Dutch adults for 2 years with some extra domains like stress and sleep. We did this on 5 sites in the Netherlands, and the Wageningen site, who is in charge of the mind diet, is also here. We started this 2 years ago, so we're now very busy collecting data. We have another 5 years to interpret all the data. In the next work package with data scientists, we would really like to see, okay, so who is actually benefiting from this lifestyle intervention, and can we find predictive markers of this intervention? In a more mechanistic work package with neuroimaging, where I'm scientifically most involved in, we would like to see, okay, are these markers now biologically relevant? So what happens in the brain, and does this happen through the gut immuno-brain axis, perhaps? And then in the 4th work package, we would like to use those predictive biologically relevant markers that come from the earlier work packages in an engaging, personalized e-health tool, where we really look at, can we give personalized feedback that's really motivating, that people can sustain their lifestyle change, and also using behavioral change strategies. So a bit more on this mechanistic work package. By the way, if you want more information also about our partners and our plans, please look at our website. So we have multiple interventions in this mechanistic work package. One of them is also a multi-domain lifestyle intervention, a bit similar to the finger-NL intervention. We have a mind-type diet, we have physical exercise, we have mindfulness or stress reduction, we have a sleep training and a cognitive training. And this specific multi-domain lifestyle intervention, because it's more mechanistic, is in way less participants, but also way shorter, only half a year. And we have a high intensity and a low intensity group, and we test them before and after on lots of things. We have neuroimaging, a neuropsychological test battery, fecal blood and urine sampling, and a smartwatch. And so our primary outcomes are really functional neuroimaging measures. Functional MRI during an MBAC working memory task, cerebral blood flow with arterial spin labeling. It was mentioned yesterday, so it's really hard to measure neuroinflammation. Of course, we're interested in it. There's not so much done, so it's not our primary outcome, but we do want to look at it using quantitative imaging like QSM, looking at iron deposits, and also doing magnetic resonance spectroscopy, looking at things like myoinositol. Then on the gut immuno side, we collaborate with Wageningen University. We look at the blood information markers and also gut microbiome markers in stool, in urine, and in blood. And here, I would like to stress, we really want to look at function with metagenomics and metabolomics. So, to conclude, I hope to have shown you that brain functioning relates to motivation and cognitive control, and it can be modulated by nutritional, inflammatory, and gut microbiome factors. And future nutritional or dietary or lifestyle interventions like MOCIA should reveal whether the intervention-induced changes in these microbial and inflammatory markers can actually affect brain function in aging, and also the motivation to actually sustain a lifestyle change. Thank you.